So we are here in French Guiana, a region hosting a huge biodiversity. For instance, if we look at freshwater fish, you could see in only one side of a French Guiana river almost the same species number as you could ever see in all rivers of mainland France. Nevertheless, this biodiversity is threatened by a lot of human activity, such as deforestation, dam construction, or mining activity. So we need to evaluate the impact of this human activity on biodiversity. There are a lot of different methods to monitor biodiversity, but I just use this tiny suitcase that I'll bring here to monitor biodiversity. So this suitcase is in reality a water pump, and thanks to a filter, I can collect DNA released by organisms such as fish in the water, and then I can connect DNA to its corresponding species. This method is called environmental DNA and has been used for now 10 years in our lab. And several studies have already shown its efficiency in French Guiana. But there is still some challenge that need to be addressed, and now the objective of my PhD. So the first challenge is to switch from the French Guiana scale to the all Amazonian scale. And this is a real challenge because using environmental DNA implies to know about all species DNA, and we don't have access to this data for all of the countries of the Amazonian scale. Then the second objective of my PhD and the second challenge is to switch from a spatial approach, that is to say sampling several sites on the same day, to a long-term monitoring approach, implying the sampling of the same sites, but at different times of the year. And this is also a challenge because between the different sampling events, you have some water parameters that are changing, such as the water level, the water flow rate, or the water temperature. And all of these parameters can impact the amount of DNA that you collect. Then the main interest to switching from a spatial approach to a long-term monitoring approach is to be able to assess population dynamic, to be able to take action for species undergoing a decline. But until now, environmental DNA is only interpreted in terms of presence-absence information, because there is a lot of gases between the number of species in your river and the number of DNA that you collect. So to conclude, pursuing the development of environmental DNA is especially important in tropical systems because for now it represents only a small portion of literature while facing the highest biodiversity loss.
Thanks a lot. Yuchen. Thank you. This one?
You use this one, but I need to find the
Yeah, it's this one. Yeah. It's okay. So hello everyone. My name is Bai Yuchen. I come from French Institute in Rare and AMAP works. So AMAP, sorry. So today I will introduce my PhD topic. The topic is focusing on the leaf word separations and the leaf area estimator. So the first part of my PhD thesis is working on the leaf wood LiDAR data separations. My three supervisors and me, we have working a lot on this topic for 2 years. So it's really my pleasure to share the first topic with you because we just published a paper about it and it get accepted by one of the best AI conference. So for the leaf word separations, we have proposed one special split schema we provide a special split manner to divide the tropical forest LiDAR data into meaningful subsites. So we use the AI model to do the classifications on it. And the second part of my PhD topic is more about on the evaluating and ranking the uncertainty and bias of the currently used live area density estimators. So we consider a lot about the voxel size, about how to analyzing the LiDAR data, about the leaf angle, density of angle, and position of leaves. So we working on this in using simulation data. Generally, we already have all the true factors, all the true results about the simulation data. So we can precise it. We can do some post processing about it. Okay, yeah. So thank you.
Thank you. Next, Vincyane, please.
We know that trees need light, water, and nutrients. We know that any species of trees cannot establish itself anywhere. And we know that trees interact with the rest of the living world. They are the product of their parent trees. As a seed, they can be dispersed by an animal. And once established, they had to survive pathogens and neighboring plants. Which of these factors are the most important to determine the spatial position of each species? All cool stochastic events explain more their distributions. Moreover, the tree needs can evolve during its development. How do I answer these questions? For the abiotic environment part, many environmental characteristics are linked. Topography influences moisture and soil characteristics. Light influences temperature, and temperature influences moisture. So topography and light can be sufficient proxies. Great, there are new tools for carrying both. LiDAR, which is a laser that scans the forest in 3 dimensions. To check that LiDAR is indeed sufficient to provide information on temperature, moisture, and soil characteristics, I've installed climate sensors and taken soil sample on my study site. And this month, I will take a walk with my backpack with the LiDAR in my backpack to scan the structure of the forest and deduce the light intensity at each point and see if LiDAR products match climate sensors and soil sample. For the biotic environment, we have the chance to have a super complete inventory of trees above 1 centimeter diameter, with every information we need: botanical identity, size, and spatial coordinates of each species or of each tree. So I know where the parents live and who the neighbors of each tree are. Then I will pull all collected information in models and explore each species needs across its different life stages. What for? Knowing the environmental needs of trees during their development will help to improve forest dynamics models and thus better predict the fate of trees in the face of climate change. This will also be useful for local forest managers to adapt management of French Guiana's forest. Thank you very much.
Thank you. Julien will be the last.
Hello. Today, I'm going to introduce you a tree. Surprising in French Guiana? Not really. But it's not just an ordinary tree. It's l'angélique in French. Dicorinea guianensis for the scientific community or for few people fluent in Latin, and Angela for his close friend. It's been over a year since we became friends with Angela, or enemy, depending on how many cups of coffee I drink. But why is this species is not an ordinary tree in French Guiana? The answer is easy. 54%. Dicorinea guianensis account for 54% of the timber harvested in French Guiana. That's a lot of pressure on a single tree species, and that's a pressure that's going to increase in the coming year. Between the huge human demographic growth in French Guiana and the forestry targets set by the Office National des Forêts that are ambitious but necessary to provide for the territory's need, and to add insult and injury, climate change uncertainty, and period of intense drought. To cope with this challenge, I explored two potentially useful solutions in my thesis. As a first step, try to predict the response capacity to climate change of the species harvested by studying the DNA of a large number of trees of Dicorinea guianensis. I'm trying to find out if the trees that are most frequently subject to water stress have signs of adaptation in their DNA. To do this, I work on a whole genome scale of individual. Additionally, I also study the dispersal process of the species in face of environmental variability by using microsatellite genetic mark. As a second step, and to cope with future challenge in forestry, we can diversify the harvested species to reduce the pressure on a small group. In French Guiana, 5 types of wood account for 3/4 of the wood harvested in volume. But before starting to harvest new species, we need to know more about them to minimize the impact on the local population of this new species. That's another part of my thesis. I work on a group of species called Mao Noir. Different species of Mao Noir share many similarities in their botanical feature, but also in their DNA. And I'm trying to bring order in our knowledge of this group. It will be unwise to exploit unknown rare species that are already at risk of extinction. To put my thesis in a nutshell, we could say I'm trying to find information about trees in their DNA to minimize the impact of forest harvesting in French Guiana.